Continuing our trend of gram-negative pathogens, we now have the genus Vibrio. These, however, do not belong to the Enterobacteriaceae. Vibrio species are gram-negative, facultative anaerobes, and they are oxidase positive. Remember, the Enterobacteriaceae are oxidase negative, which allows you to distinguish the Enterobacteriaceae from other gram-negative, especially rod-shaped bacteria. The textbook talks about two different genera together, both Vibrio and Aeromonas. Again, because of the lack of time, we can't cover everything. Vibrio is far more common, so that's the genus that we're going to cover. But in the textbook, you've got Aeromonas, so you can always look at that for your own personal reference. Vibrio species do fermentation of various carbohydrates. They have polar flagella and are often associated with contaminated water. There are two major types of infections caused by various Vibrio species, either significant GI disease or they are also causes of flesh-eating infections. They have this kind of curved, rod-shaped bacterium, sometimes called comma shape, and you can see the polar flagella here, meaning they are motile. They can move through, especially water. They can grow anywhere from 14 to 40 degrees C, and they do require salt. They're considered to be kind of halotolerant or halophilic. This is why you often will find Vibrio species in warm marine waters. One of the more common areas in the US to find Vibrio species is in the Gulf of Mexico. The water there is very warm, and of course it's salty. They will tolerate a pH anywhere from 6.5 to 9. They are actually very, very susceptible to stomach acid. So the question I'd like you to think about for just a minute is, how do they actually cause infection? So even though they're highly susceptible to stomach acid, what we generally see is that they can be at very, very high numbers in the water or in a contaminated food item. And so a patient is often ingesting lots and lots and lots of bacteria, some of which can escape the stomach. The other issue is that Vibrio species are often very common in areas where patients tend to be more malnourished. When you have someone who's malnourished, the pH of the stomach is raised, again, allowing the Vibrio to survive. Finally, Vibrio can also be associated with things like contaminated shellfish, and the buffering provided by eating the bacterium with a meal can also help raise the pH of the stomach temporarily. The majority of Vibrio species, like I mentioned, do have a polar flagellum. They also have pili to help them adhere better, and those that have a pilus tend to have increased virulence. In Vibrio cholerae, the species that causes the disease cholera, they have what's called a toxin co-regulated pilus that really helps them adhere to the gastrointestinal tract. The three species we'll talk about are Vibrio cholerae, Vibrio parahemolyticus, and Vibrio vulnificus. You'll see the clinical disease on the right-hand side of this table, and you'll see that all three of them can cause bacteremia. Both Vibrio cholera and Vibrio parahemolyticus can cause gastroenteritis, and parahemolyticus and vulnificus can both cause wound infection. However, most commonly, you'll see cholera associated with gastroenteritis and Vibrio vulnificus with wound infection. That's just what we tend to see most commonly. They, as gram-negative organisms, do have LPS. So remember, lipid A, the core polysaccharide, and the O antigen. The O antigen is used to determine serotypes or serogroups. They have a polysaccharide capsule, except for Vibrio cholerae strain O1. And unlike most bacteria, Vibrio species do have 2 chromosomes. So this is pretty unique, and which is why we generally say most bacteria have a single chromosome, but not all. So they have 2. It is also common for Vibrio species to carry plasmids that can confer antibiotic resistance. Here they give you the different species and some of the various virulence factors that the organisms have. We're going to talk about cholera first, so we'll look at those virulence factors. Cholera toxin, which leads to hypersecretion of electrolytes in water. The toxin co-regulated pilus, which serves as a binding receptor for a bacteriophage that carries the toxin, and also helps to mediate bacterial adherence to intestinal mucosal cells. And other adhesion factors, other toxins that are, here you see accessory toxin, increases fluid secretion. This zonula occludens toxin, which increases intestinal permeability, and neuraminidase, which also modifies the structure of the eukaryotic cell to allow increased binding sites for cholera toxin. So this is a pretty nasty organism that is really well evolved to do what it wants to do. Vibrio parahemolyticus has a hemolysin, which acts as an enterotoxin to induce secretion. Vibrio vulnificus has a capsule, which is antiphagocytic, and lots of enzymes, including cytolysins, proteases, and collagenases to mediate tissue destruction. So it's not a genus that we talk about a ton, but it's a genus that can cause some really nasty complications. The cholera toxin is an example of an AB toxin. There are 5 B subunits that bind to the host cell. The A subunit, instead of interfering with protein synthesis like we've been talking about with the Shiga toxin, with diphtheria toxin, in this case, the A subunit gets into the host cell and it leads to the formation of cyclic AMP. When the cell has too much cyclic AMP, that alters the ion balance in the cell, which leads to a hyper release of water and electrolytes. And that water will flow from the intestinal cells and from the blood vessels surrounding the intestinal cells into the intestinal lumen, which is why you get a copious volume of watery diarrhea. Now, if the bacterium is not able to adhere, either through the toxin co-regulated pilus or through those chemotaxis proteins, there is no infection. So Vibrio cholera needs to be able to bind to the intestinal lining in order to establish infection. If it just washes through, no problem. There are many strains of Vibrio cholerae which do not produce cholera toxin. We saw the accessory cholera toxin and the zonula occludens toxin. Those 2 toxins, the accessory cholera toxin and the zonula occludens toxin, will also lead to diarrhea. However, it is the production of cholera toxin that leads to the copious, deadly diarrhea. So even without that toxin, this bacterium can still cause gastrointestinal issues, but the strains that have this toxin are much more dangerous. The O1 and O139 strains are actually the only ones we know of that currently produce the cholera toxin. So all of the other strains of Vibrio cholerae can cause diarrhea, but they don't produce cholera toxin. The most recent pandemic, which was back in the late 60s, early 70s, was caused by a biotype of the O1 strain called L-Tor. Now you'll notice we're using a new term here, biotype. We've used serotype or serovar, which are basically interchangeable. When we use serotype or serovar, what we're talking about is the antigenic profile of the organism. So a particular serotype will produce a particular type of immune response because it has particular antigens. A biotype is basically just a group of organisms that are genetically identical. And so there are a handful of biotypes that are still all considered the O1 strain. Because remember, the O antigen is simply part of the LPS. And so there are a number of different organisms that all contain the O1 version of LPS. And so one particular group of them, the L-TOR group, is what caused the most recent pandemic. However, we still do have cholera circulating around the world to this day. There was a major outbreak in Haiti in 2010 after the major earthquake they had there. There is an ongoing outbreak in Yemen. There have been periodic outbreaks in parts of the Middle East and sub-Saharan Africa. So this is a disease that we still see worldwide and is still a cause for concern. Vibrio species grow in marine environments and estuaries. They need that saline environment because they have to grow in salt. They flourish in warm, salty water. So again, places like the Gulf of Mexico, all of the equatorial regions where the water is nice and warm. They are also found in shellfish. So I'm a vegetarian. I don't like to eat meat anyway, but like raw oysters is one of those things that I'm just like, dude, what are you doing? That's just like a cholera factory. Depends on, I guess, where the oysters were raised, but I would definitely never eat oysters or any sort of shellfish raw if it was from any sort of warm water environment. They are spread through contaminated food and water. Like I mentioned earlier, because we have that sensitivity to the stomach pH, it generally requires a very high inoculum to cause gastrointestinal infection. This is usually found in areas with poor sanitation. Cleaning the water supply will prevent Vibrio infection for most of the patients, and it will also prevent contamination of local warm water. What we tend to see is that we have high levels of cholera in areas where people use the same water supply for drinking, bathing, and cooking. So here's our clinical summaries. Cholera begins with an abrupt onset of watery diarrhea and vomiting that can progress to severe dehydration, metabolic acidosis, and hypokalemia, and hypovolemic shock. Hypokalemia just means low potassium, so it's because of that secretion of water and the electrolytes, which are your ions, basically. We can also have milder form of disease if we are toxin negative. Vibrio parahemolyticus can cause a self-limiting gastroenteritis with an explosive onset of watery diarrhea. Isn't that a great description? Explosive onset of diarrhea. Vomiting, cramping, headache, fever. Wound infection is associated with exposure to contaminated water, but more commonly causing a wound infection is going to be Vibrio vulnificus. These are severe, potentially fatal infections characterized by redness, pain, swelling, necrosis, and septicemia. So here's cholera. This is a headline from a few years ago, more than 500,000 infected with cholera in Yemen. The outbreak in Yemen is now associated with over 1 million cases, if I'm not mistaken. What you have in the top left is an image of what are called cholera cots. So the diarrhea has a relatively abrupt onset, and if the patient doesn't die, the patient will recover in 3 to 4 days. But that's if the patient doesn't die. The idea is you have to keep your patient alive long enough to clear the infection. Our bodies are actually pretty good at getting rid of cholera. Because of these copious amounts of diarrhea, the bacteria often just get flushed away. So you just have to flush them all out. But because you can die of diarrhea, you could die before you can clear out the infection. So you'll see these cots, they have a hole in them. There's a bucket that goes under the hole. I'm hoping by now you've been able to picture what part of a person's anatomy would go above the hole. The patients are laid down on the cholera cot. The fluid that is passed is collected in the bucket. This is called rice water stool. The patient experiences so much diarrhea at such a rapid rate that after the first few times, there's no more food in there. There's no real feces. So it's just water, bile, and mucus. And this is just teeming with bacteria. Huge numbers of bacteria. So the clinicians will measure out the volume that the patient has passed. And then they do, you can see it here, it's called oral rehydration therapy. You just keep the patient from getting too dehydrated. And it sounds silly, but this is actually what they do. You can Google it, cholera cots. Now, Vibrio vulnificus causes really significant wound infections. So these are headlines from a few years ago, but you'll notice Mobile County, so Alabama, right along the Gulf of Mexico, 6th case of Vibrio. Florida resident catches deadly ocean-borne bacteria, again, Gulf of Mexico. Man dies after flesh-eating bacteria infects new wound. So he got a tattoo, right, which is a wound. It's a needle being poked into your skin. I got no problem with tattoos. Got a couple of my own. No judgment, but it's wounds. And then he got in the water off Galveston Island, which is in the Gulf of Mexico, outside of Houston, and he got Vibrio vulnificus infection and died of septic shock. And so you can see the redness and the swelling in the tattoo. You may remember a few years ago, Hurricane Harvey, that massive hurricane that really caused significant flooding and damage to Houston. That water, those floodwaters were warm, right off the Gulf of Mexico, full of bacteria. And there was at least one patient who died of a Vibrio vulnificus infection because of the floodwaters. So I love the Gulf of Mexico. It's warm. I love Texas. I did my PhD and postdoc there. I'm actually going to Texas in a little bit. But do not go into the Gulf of Mexico if you have any sort of wound. Flesh-eating bacteria kills Virginia resident. Same thing, Vibrio vulnificus. This one I thought was interesting. Public health investigates rare infection linked to fish in supermarket tanks. This was in Washington, so in King County. A man was diagnosed with a bacterial infection caused by Vibrio vulnificus after consuming fish purchased from live fish tanks at a local store. And so because of the raw fish, it can get into the wounds as you're on your hands as you're preparing the fish. So just be careful. Here's even more. A woman dies after contracting Vibrio in Nueces County. This is this is Corpus Christi, Texas, which is also on the Gulf of Mexico. Vibrio bacteria death shows why you should beware of raw oysters. Vibrio outbreak linked to crab meat imported from Venezuela. So again, not an uncommon infection, and it can be very, very serious. So these are reports of Vibrio illness in the United States up through 2016. And so this is Vibrio parahemolyticus, which this is going to be mostly gastrointestinal. This is Vibrio vulnificus. This is total Vibrio infections. So some of these are probably cholera. Flesh-eating bacteria is infecting more people and spreading to new areas. Scientists blame climate change. So sometimes when I teach a class like this, the majority of students are interested in clinical laboratory science, interested in going into medicine, and they will sometimes express to me, I'm trying to think of a nice word, their dissatisfaction with having to take classes like ecology. But what they're missing is the connection between ecology, our ecosystems, our environment, and infectious diseases. So with climate change, with warming global temperatures, you can see spreading of organisms like this. And we'll talk about other types of organisms that are also spreading because of our changing climate. So it's really important that you consider how all aspects of biology, including ecology, integrate into human health. Vibrio are very sensitive to drying and acidity. So, if something was contaminated and then left out to dry for a while, it would be fine. Cultures must be done rapidly on appropriate media. So remember, it prefers a hypersaline environment, like a marine environment. So your growth media has to be supplemented with saline. Antibiotic treatment is not usually prescribed because as long as your patient can survive the diarrhea, they're going to be able to clear that infection. Instead, we would do fluid replacement therapy. You have to measure how much is coming out so you know how much to put back in. And sometimes the volume going out can be something like 15 liters a day. So just think about that volume of fluid coming out of your body in a single day. How awful that would be. This is nothing like you ate something funny and are going to be in the bathroom for a day. This is liters and liters and liters of fluid just streaming out of your body. Vibrio vulnificus, however, would require combination antibiotic therapy. Remember, Vibrio species can carry plasmids that confer antibiotic resistance. We don't want to mess around with a Vibrio vulnificus infection. This can be fatal pretty quickly, so we want to prescribe at least 2 antibiotics with different mechanisms of action to help treat our patient's infection. There is an oral cholera vaccine available in other countries. Again, debatable efficacy, and the frequency of patients in the US getting cholera is very, very low. However, I do want to tell you, cholera is in the United States. So I worked during my PhD next to a lab that studied cholera and Francisella, which we haven't talked about yet, but we will. And they would periodically go from San Antonio to the Rio Grande Valley, so about 4 hours away. That's the border between the US and Mexico, is the Rio Grande, the river. They would take water samples from the river, and almost always they would find cholera in the Rio Grande. So we have cholera in the United States. It's just a matter of appropriate sanitation and appropriate food preparation that keeps it from just kind of exploding in this country.